Well, hello everyone, and welcome to lecture 9 in colloid and surface chemistry. So today, what I would like to cover is the general structure of lipid molecules and look at some ways in which the self-assembly of lipid molecules is somewhat similar to surfactants, but because of their structure, they yield slightly different self-assembled structures. And I would like for us to think about ways in which we can prepare in the laboratory some of these self-assembled assembled lipid systems, and possibly think of some applications of these self-assembled lipid systems, because they're very, very important in colloid and interface science. So lipids then are defined as molecules that are soluble in non-polar solvents. So have a think about what that means for a moment. It means that they're generally not soluble in water, and the reason really why they're not soluble in water is because they have very, very long alkyl chains, and that renders them completely insoluble in water. Sometimes they don't only have sort of one long alkyl chain, but it can be that they have multiple alkyl chains, which of course renders them even less soluble in water. And there are many, many different types of lipid molecules, many different types of lipid classes, but the three most important classes of lipids for this module are the triglycerides, the phospholipids, and the steroids and waxes. And we're going to try and have a look at the applications of these lipid systems today. So we are going to focus very much on the phospholipids, because these are the lipids that make up the bulk of our own cellular membranes in mammalian cells. So they're very, very important from an academic perspective. So you've seen on the previous page there some generic lipid structures. We've spoken about some properties of lipids already. Have a think about what is the difference between a surfactant and a lipid. So have a look back over the lectures that I gave on surfactant systems and see if you can work out some key differences between lipids and surfactants. And ultimately it comes down to solubility. So the phospholipids then are one of the most important classes of lipids in biology. As I said, they make up the bulk of our mammalian cell membranes, but they're also very, very important in colloid and interface science. Not least because when we probe the self-assembly of phospholipids, we can use them as models for the cell membrane, and we'll talk about that in a little bit more detail as we progress. That's one of their main applications in colloid science. But have a look at the generic structure of a phospholipid here on the left. It contains a long alkyl tail, usually in the form of some kind of fatty acid, usually a bulky non-polar tail then. And as you move into the head group, it has a hydrophilic head, usually has some kind of glycerol linker that links a phosphate group and some other kind of polar group. So there's a generic structure on the left there. You can see they very much resemble surfactant molecules, right? There's that hydrophobic portion of the molecule and a hydrophilic portion of the molecule. That's just like a surfactant. It's just that the alkyl tails in lipids tend to be much longer. They're of a higher molecular weight, and they tend to have bulkier non-polar groups, which render them insoluble in water. And because of that, their self-assembly in terms of the structures that they assemble into are slightly different. So have a look at the diagram on the right-hand side of the slide now. There's a column which shows you the basic phospholipid structure. You can see that long alkyl tail group. You can see a head group region that's linked to the alkyl tail group through this glycerol group. There's then always a phosphate group. That's where the term phospho comes from in phospholipid. And then there's an X region in the first column, and that shows that there can be multiple different head groups for phospholipids. So you can get anionic phospholipids, you can get zwitterionic phospholipids that have both a positive and a negative charged head group region. You can get you can even get cationic phospholipids, but the most relevant phospholipid is one called the phosphocholine group of phospholipids, and they are zwitterions. They have a phosphate group followed by a quaternary ammonium group. And the reason why they're the most relevant is because you find them in high concentrations in the mammalian cell membrane. But lots of different phospholipids. Now, as I said, they make up the mammalian cell membrane, and this is an example of how phospholipids self-assemble. They self-assemble in this instance into something called the phospholipid bilayer. And the phospholipids here are shown with a yellow ball representing the head group and a brown tail representing the tail group. And they sandwich together in this bilayer-like structure. And that's a thermodynamically favourable arrangement because most of the solution, if you like, in our body is aqueous. They have to exist in an aqueous environment, and this is the most thermodynamically stable way of arranging a lipid on this scale, to have those long alkyl tail groups pointing inwards and interacting with each other, and having the charged head group, polar head group, facing out towards solution. Of course, that's perfectly happy to interact with a water phase. So a first important consequence of lipid self-assembly is inherent in our own biology. Now, one very interesting thing they do is they, of course, self-assemble on the surface of water in much the same way as surfactants do. It's just that they're not soluble like surfactants. So when you have a very large hydrophobic group within the molecule, as you do within these lipids, those lipid molecules are themselves rendered insoluble in water. And what they do is they form an isolated two-dimensional monolayer on the surface of water. So this is a single molecule thick layer of lipid on the surface of water. The head groups are solubilized or are in solution, and the tail groups point away from solution. And this kind of film on the surface of water, and it is a film, compare that to a surfactant. It's a much denser film than a surfactant because of the molecular weight of these lipids. Those films are called LANGMUIR monolayers, and phospholipids are very good examples of molecules that form these LANGMUIR monolayers. And fortunately, there are tools to be able to study the properties of these LANGMUIR monolayers. And the most useful tool is to use something called a film balance or a LANGMUIR trough. And you'll see in the schematic diagram that I've drawn there, what you do is you dissolve your lipid in a solvent in which they're soluble. It's usually a very volatile lipid, such as chloroform. And you drop that solvent containing the lipid molecule onto the surface of water. And you wait for the solvent to evaporate. That's why it's got to be quite a volatile solvent. And once the solvent has evaporated, you are left behind with this single film of lipid molecules on the surface of water. And these are all Teflon troughs that you conduct these experiments in. And you're able to move the Teflon barrier, you can see in the image there, from left to right, and you can compress and expand that film of lipids as you do so. When you compress or expand the film of lipids, what you're actually doing then is changing the area that's available to each molecule. We call that the area per molecule. As you decrease the area available to them, you're decreasing that area per molecule. And what happens is there are changes in a property that we call the surface pressure. And as you compress them, the surface pressure starts to increase. And we'll talk about how that increases and why that increases in a while. But the surface pressure then is monitored by something called a Wilhelmy plate, and you calculate the surface pressure. Nowadays, of course, this is all done by computer, and you just get a value for the surface pressure from the software you're using. But the correct calculation for the surface pressure is to take the surface tension of water in the absence of the lipid and the surface tension of the water in the presence of the lipid, and of course that is a surface pressure. So you can measure the lateral film pressure of as a function of the molecular area, and what you gain is this surface pressure area isotherm. So this is for a lipid system, and what that does is it tells you information about the structure of the monolayer and any so-called phase transitions that occur. So let's have a look at that diagram on the left there. It's a graph of surface pressure against area per molecule, and we will read it from right to left. So we are starting with the barriers of our Langmuir trough open, so that the lipid molecules on the surface have lots and lots of room to wander about. That's on the far right-hand side of that diagram. So we would say that that is in the gaseous state. Okay, all of the lipid molecules are moving around much like a gas, but in 2 dimensions. As we compress the monolayer, the lipid monolayer, as we move the barrier closer together, then those lipid molecules start to interact. They're no longer in a kind of gaseous-like state, and suddenly they get close enough together to start to start to interact. And they interact through these long alkyl tails kind of reaching out and touching one another. And because of a kind of force that is involved between those interacting lipid molecules, a repulsive force, the surface pressure starts to go up. Now, those lipid molecules start to interact even further, and they enter what's called a phase coexistence region, and they start to align themselves with respect to the surface plane of water. So they start to all point in the same direction, perpendicular to the plane. And here you've got a mixture of phases, and that plateau in surface pressure that you can see is a phase transition. Eventually then you work your way through that phase transition, and the surface pressure starts to go up, and now you've got all of those lipid molecules with their tail groups pointing away from solution, perpendicular to the interface, and they have entered this so-called liquid condensed phase. Now eventually you'll run out of room on that surface, and your monolayer will collapse, and you see that with a decrease in surface pressure then. And essentially what happens here is that your monolayer buckles, and it starts to form 3-dimensional structures in what we call the subphase, the water phase beneath it. Okay, there is an explanation of each of those phases there if you're interested. I think I've said everything on that slide though. So when you run out of room on the surface, then the monolayer actually collapses, and the mechanism of this is unclear, and it's still debated in the literature. But it's very likely that the monolayer starts to essentially buckle, and it probably starts to form some of these kind of bilayer structures or multi-layer structures in this aqueous subphase. And you can see here, this is some images taken with something called a Brewster angle microscope. And these allow the direct visualization of these nucleating liquid condensed phases. So you can see that I show you some images of these nucleating liquid condensed phases from a Brewster angle microscope at different surface pressures. And the shape of these is governed by the molecular structure of the lipid molecule. There are some quite striking effects that can occur when these Langmuir monolayers interact with ions in solution. And some investigations have been done that look at the interaction of small cations, usually sodium and calcium, and how they interact with phospholipid Langmuir monolayers. And this is really important because that Langmuir monolayer is essentially a model for the cell membrane. It's as close a model as we can get. We can't replicate the cell membrane, but it's as close a model as we can get to studying what happens when we put various things in the vicinity of the cell membrane. So it's a very good way of studying a model of the cell membrane. For example, if you wanted to know if a particular drug molecule penetrated the cell membrane, then you could use a Langmuir monolayer as a model for the cell membrane, and you could measure the penetration of a drug molecule through that Langmuir monolayer, and that would give you a good idea of whether or not that drug molecule would penetrate the human mammalian cell membrane. Now, they adsorb on the surface of water, but they also adsorb at the solid-liquid interface. And there are a number of ways of immobilizing these monolayers and bilayers upon a solid support. And what you want to do first is take either a silicon or a gold substrate and chemisorb a self-assembled monolayer onto those substrates. And your aim here is to create a very dense hydrophobic interface that the phospholipids can interact with. So the first thing you've got to do is stick down what we call a self-assembled monolayer. Then you can deposit the phospholipids directly onto the surface using a number of different techniques. You can use a structure that we call a vesicle, and I will talk more about these towards the end of this session. And you can basically rupture the vesicles over the surface and form a phospholipid monolayer. Or you can take a Langmuir monolayer and you can dip a block of silicon or gold into the Langmuir monolayer, and you form you transfer the monolayer onto the solid support. And you can work out how much of the phospholipid you've transferred onto your surface. And you can see there that you can get a range of different types of phospholipid films. You can get monolayers and you can get bilayers. Of course, bilayers are a great model for the actual human cell bilayer, so a lot of interest goes into generating these kinds of surfaces. You don't have to dip them vertically, you can also dip them horizontally. Slightly different technique, it's called the Langmuir-Schaefer technique. And there are lots of different ways that you can study the structure of these films. Now, we're not going to talk about this. I'm not going to talk about this in too much detail here because I've got a lecture towards the end of this section. We'll look at it next time probably about techniques in colloid and interface science. So I'm not going to talk about this right now. These are two techniques that are very commonly used to study the surface, the film structure on the surface, and we're going to go into a lot more detail about these techniques in the next session. Similarly here. Now, as I said, the key application of these LANGMUIR-BLODGETT films, or these LANGMUIR monolayers, is that you can use them as a model for the cell membrane. And there are lots of different lipids that can form LANGMUIR monolayers. It's not just phospholipids. These are a class of lipids called the sulfur betanes, and they also form LANGMUIR monolayers. Okay, so I just want to have a quick chat about vesicles, and because this is another structure that phospholipids self-assemble into. So firstly, what is a vesicle? Well, these are particles that are spherical in shape, and to be a vesicle, it's got to have at least one lipid bilayer. So you can see a phospholipid bilayer there. They are usually composed of phospholipids and steroids, and they range in size from anything from 50 to 800 nanometers. And again, the membrane structure is analogous to that of the mammalian cell, even closer to that of the mammalian cell than a LANGMUIR monolayer, because vesicles are curved. So I show you there an image that highlights the difference between a bilayer or a monolayer, a micellar structure, and a vesicle, or a liposome is another word that you will see for a liposome, for a vesicle. So you've got a hydrophilic core, you've got a water core, and then a bilayer structure. So they were discovered in the 60s, and they're really important structures because they've got lots of different applications. One of the major applications of vesicles nowadays is in drug delivery, because you can incorporate a drug either into the aqueous core of the vesicle or into the bilayer of the vesicle. And of course, that's very flexible because you might have a hydrophilic or a hydrophobic drug, and you can deliver these things to the target. You can molecularly functionalize vesicles as well, so that you can think about targeted delivery of drugs to a specific spot in the body. And you make them quite easily. There's a really cool schematic on the right hand side of this slide there that shows how phospholipids self-assemble into a vesicle. That's a molecular dynamics computational study. But they're very easily prepared in the laboratory. What you do is you take your powdered lipids and you dissolve them in an organic solvent. It's usually chloroform. You remove all of the organic solvent, so you have a lipid film left on your round bottom flask. You then rehydrate the lipids in a buffer, usually a tris buffer or a sodium chloride buffer or phosphate buffer. You agitate the solution, and the vesicles form as the water essentially removes lipid from the wall of the vessel. If you do it that way, you get these things called large multilamellar vesicles. You can then sonicate that system, and you end up with small unilamellar vesicles. And as I said, you can use them to form LANGMUIR films. You can deposit vesicles onto a solid surface, and they will form monolayers and bilayers dependent on the chemistry of your solid surface.